This is a run through of the Unified paper that was produced by the OCR exam board for their A-level biology, which was going to be examined for the first time in June 2017. So this starts with a question on a respirometer. So they tell you that a group of students set up a simple respirometer, as shown in figure 1.1, used it to determine the rate of respiration in germinating mung beans. Small muslin bag of soda lime into the syringe added 5 germinating mung beans, held in place with the syringe plunger. The students measured the movement of the red fluid in the capillary tube. After each set of readings, the plunger was reset to return the fluid to its original position. So first thing when you get an apparatus like this is to understand what's going on. Hopefully you've used one in your lessons, but if you hasn't, then here you have a germinating mung bean, and the mung bean is going to be taking in oxygen. It's going to be respiring aerobically, and the oxygen is going to be converted into water because it's the final electron acceptor in the electron transport chain in the mitochondria. Now, the germinating mung bean is going to be using glucose, and it's going to be producing carbon dioxide in link reaction and the Krebs cycle. Now that carbon dioxide is going to be absorbed by the soda lime. So any change in volume that occurs into the barrel of the syringe, so any change in volume that occurs here is due to the consumption of oxygen being turned into water. So as the oxygen is turned into water, the pressure will fall and the fluid will move in this direction. So when they're measuring the movement of that red fluid, they're measuring the uptake of oxygen by the mung beans. So here are the results, and in a results table, 3 replicates, and then one limitation of using this method to investigate respiration rate. So when they're looking for a limitation, they're expecting you to criticize the apparatus and the use of the apparatus. So the suggestions that they have here are the idea that, zoom in a bit, oxygen will leak from the connectors, into the apparatus, so that will reduce the movement of gas. Oxygen uptake may not be a good representation of the respiration rate in the germinating seedlings, and that's because the germinating seedlings may be respiring anaerobically if they've been soaked in water to get them to start to germinate. Additionally, why there may be a limitation is that you're only measuring a small volume of gas. The measurements are only taken every 30 seconds, and it's difficult to read the meniscus. So it may be subjective due to the action of the experimenter. So it now says, read the procedure carefully, identify one variable that had not been controlled in this experiment, and suggest an improvement to control that variable. So now you need to think about what is a variable that they didn't control that could affect the independent variable. Now they're trying to measure the rate of respiration, so you need to think about any variable that they haven't controlled that might affect the rate of respiration. So things like the mass of the seeds wasn't given, you should measure the mass of the seeds at the start. The mass of the soda lime wasn't specified. The size of the syringe is not given. Additionally, they were looking for, they didn't specify the tube, the diameter of the capillary tube. Therefore, you could improve it by using a specified length. And a specified internal diameter, so a 1 millimeter lumen. The answer I'd expect most of you to go for was that the temperature is not controlled. And why that's important is that's going to affect the rate of respiration, because respiration is an enzyme controlled reaction, and also it's going to affect the volume of the gas. Therefore it's going to affect the movement of the red liquid. So the improvement would be allowing the apparatus to stabilize or equilibrate before you take the readings. On to describe how you would add the red fluid to the capillary tube at the start of the experiment. Well, for this they were just expecting you to say that you would dip it into a small beaker and allow that fluid to run into the capillary tube. The data shows an anomalous result at 60 seconds. Explain why the result is considered to be anomalous. That's the first part of the question. And then the second part of the question is describe one correct way of dealing with this type of result. Now this is a useful thing to read in a practice paper because it gives you an idea of what they would consider anomalous for all future papers. So if you ever came across a question that asked you about identifying anomalies in future, then you should refer to this sort of answer. What they were looking for was that it was more than 10% from the mean, or it was different from the other data at 60 seconds, and you must explicitly say at 60 seconds, not that it's different from the other data, it must be at 60 seconds. Or it doesn't follow the trend for the times for replicate 3, so that it's everything within that column for replicate 3. Now what you should do about that is that you should identify it and then exclude it from the processing to produce the means. The anomaly must be identified but could be included, but you must have identified it. Or alternately, you could repeat that experiment to obtain another reading for that value. Now remember, all of these apply to future questions on anomalies. Using the data the student provided, calculate the mean rate of respiration for germinating mung beans between 90 and 150 seconds. And the answer to this one is 0.36 millimeters per second is the rate. And remember, this is rate, so whenever it's rate, it needs to be per unit time. What additional information would be needed to calculate the volume of oxygen? So this is volume. In order to work out volume, you need to know the diameter of the lumen of the capillary tube, so you can work out the volume taken up by the seed. So the internal diameter of the capillary tube is important. And what additional information would you need to calculate the oxygen uptake of this batch of seeds to be comparable with data from another type of bean? Well, to make it comparable, you'd have to know the masses that you are using for each one, so the mass of the bean seeds that you're using in each respirometer. So this is a longer 6-mark question, and they ask you to adapt the procedure used to investigate the respiration rate of a small invertebrate. So they're expecting you to adapt it in terms of altering the procedure, altering the apparatus, but using broadly the same apparatus to look at the respiration rate of a woodlouse or a caterpillar. Now they ask you to look at respiration rate, so there needs to be a measurement of time here. Then they expect you to comment on the results you might expect, so predict the results that you would see and the conclusions that you might draw from those results that you would expect. Now what they're looking for here to get the 6 marks is a clear and detailed experiment that's been effectively adapted for use with a chosen invertebrate. So you've named the invertebrate that you're going to use to allow for the comparison of the rate of respiration with that of mung beans. Now if you're going to do that, you need to know the time taken for the oxygen to be absorbed and for the red liquid to move. You also need to know the relative masses of the mung beans and of the invertebrate that you're going to use. They expect you to have a clear, well-developed line of reasoning and logically structured, and the information is relevant and substantiated. Now that will get you 5 to 6 marks. Only 3 to 4 marks, an experiment to compare the rate of respiration, but there's insufficient detail of the procedure to allow a valid comparison. Line of reasoning presented with some structure, the information presented in the most part relevant, supported by some evidence. And then so level 1 to 2 marks, an attempt is made, but little comparison with mung beans, likely to be muddled or inaccurate. So let's have a look at the content they're actually expecting you to put in here. So relevant points would be that you need to have the same mass of invertebrates and the same mass of beans, or you approximately the same mass, but you need to weigh the beans or weigh the invertebrates so you can convert it into oxygen consumption per gram of living organism. Additionally, they want you to have safe and ethical use of the invertebrates, so maybe a screen so that they can't be harmed by the soda lime that's in the muslin bag. That they want you to recognise that you need a bigger syringe because woodlouse or caterpillars are bigger than the mung beans and they tend to move around. You need to keep the temperature constant or keep the temperature the same for both assays or carry them out at the same time. Therefore, the environmental variable of temperature is affecting both of them equally. Keep the light constant or the same for both assays. Now I suspect they're thinking here that the light is going to affect the rate of respiration of the invertebrate because it's going to affect the rate at which it moves around. Use the same mass of soda lime in both assays. Measure the distance moved by the coloured red liquid at regular time intervals which enable you to give you rate, and repeat the experiment. So that was how you should adapt the procedure. The results and conclusions that they are expecting you to mention to get 6 marks is that the rate of respiration you would expect to be higher in the invertebrates because invertebrates move around and they have metabolic processes that require energy and generate waste heat. So it says that this question is about the impact of potentially harmful chemicals and microorganisms, and it starts with salts that a plant needs, such as nitrates and phosphates, are taken into the root hair cells by active transport. Now, active transport requires ATP, and the reason for that is because it's moving them from where there are few nitrates, which is outside in the water in the soil, into the plant where there are more nitrates, so it's moving them against their concentration gradient. So it says, for which macromolecule does a plant need both nitrogen and phosphorus? Well, remember, carbon, hydrogen and oxygen you need for carbohydrates and lipids. If you have carbon, hydrogen, oxygen and nitrogen, you can make amino acids. If you have carbon, hydrogen, oxygen and phosphorus, you can make DNA and RNA and ATP, which is a modified nucleotide as well. So the answer for this was DNA, RNA or nucleic acids. Flooding of fields by seawater can damage crops. Seawater contains dissolved salts, including sodium chloride. So how would flooding affect the soil water potential? Well, flooding would increase the amount of sodium chloride that's dissolved, which would reduce the solute potential, so it would make it a more negative or lower soil water potential because there are more dissolved solutes. So it would reduce or make it more negative. Then on to sodium chloride in solution dissociates into sodium ions and chloride ions. Explain, so tell us a lot about the theory, how the Casparian strip prevents these ions, sodium and chloride, from reaching the xylem of the plant by the apoplast pathway. So what they're looking for here was you to think about the nature of the Casparian strip, which is a strip of suberin, and that's going to prevent the transport of water and the ions along the apoplast pathway, which is through the cell walls. So you've got the symplast pathway where it flows through the cytoplasm. You've got the apoplast pathway where the water travels through the cell walls, hits the Casparian strip, and that waterproofing suberin forces the water into the cells, so across the plasma membrane. So that's what they were looking for you to think about explaining. So if I just show the answers, so it will ask you to explain that the Casparian strip is impervious to water. It forces the water to pass from the cell wall through the plasma membrane. Now, the phospholipid bilayer will not allow the ions or the charged particles through. So that would prevent the ions from reaching the symplast, from reaching the xylem by the apoplast pathway. Okay, plague is caused by the bacterium Yersinia pestis. The bacterium is a rod-shaped cell that's approximately 3 micrometers long. The Yersinia pestis is viewed using a light microscope with a magnification of 1250 times. What would be the length of the cell in the image produced by this microscope? So remember, magnification is going to equal image over object. Now they've told you the magnification, which is some 1250 times. They've told you and they've said that's equal to the image divided by the object size, and they've told you the object size, and that is 3 micrometers long. Now they want the answer here in millimeters. It's always best to put your calculation in the unit in which they want the answer, because I think it reduces the chances of you making a mistake. So that would be 0.003 millimeters, because that is 3 micrometers. So then you just have to multiply 1250 by 0.03, and that gives you the answer, which is 3.375 millimeters. Then it goes on to photographs taken of the image obtained by the light microscope could further be enlarged using the projector. Why might the enlarged image be unable to tell us more about the structure of Yersinia pestis? Well, this is an answer. This wants an answer about the difference between resolution and magnification. So you can magnify things as many times as you want, however you won't see any more detail because you haven't changed the resolution. So there's no further resolution when you magnify it with a projector, because there's no greater resolution than when you've looked at it with a microscope and taken a picture of that image at that point. Outbreaks of plague still occur occasionally. Plague is transmitted by several methods, including droplet infection, close contact between people, and fleas moving between infected rats and people. Now, suggest 2 ways to minimize the spread of an outbreak of plague. Now, suggest means you haven't been taught about methods to minimize the spread of an outbreak of plague. However, you should have been taught about methods of infection. You should consider, therefore, how you should minimize droplet infection, how you could minimize close contact between people, and how you could prevent fleas moving between infected rats and people. If you talk about ways in which you could minimize these factors, then you are going to minimize the spread of the outbreak of plague. Things that they were looking for were keep indoors, which would be minimizing close contact between people, increase ventilation or wear masks, which is because they've told you it's by droplet infection, measures to exclude or kill rats or fleas, and quarantining for people with the symptoms. Now, herbicides work in a number of different ways. Some herbicides mimic the action of the auxin IAA. What's the normal action of the auxin? Well, you just need to know this, that the normal action is that it stimulates cells to elongate or divide. Then they go on to talk about the herbicide atrazine. Now, atrazine works by disabling plastoquinone, one of the proton pumps in photosystem 2. Explain how atrazine would kill a susceptible plant. So this is expecting you to explain the consequences of disabling a proton pump in photosystem 2. Remember, photosystem 2 is in the thylakoid membrane, and photosystem 2 is where photolysis occurs, and that light is being absorbed by photosystem 2, and that causes electrons to move along a chain of carriers and proton pumps, and that moves hydrogen ions from the stroma into the thylakoid interior. Those hydrogen ions then flow out through ATP synthase back into the stroma from the thylakoid interior, and that generates ATP. Additionally, once the electrons travel to photosystem 1, that electron is then used to reduce NADP to NADPH, the electron and the hydrogen. Now the ATP and the NADPH are then used to regenerate RuBP in the Calvin cycle. So all of these points are relevant to explain how it would kill a susceptible plant, because if you disable plastic quinone, then you would prevent the movement of hydrogen ions across the membrane from the stroma into the thylakoid interior. You would prevent the production of ATP. That ATP wouldn't be used to regenerate RuBP. Additionally, you would stop the production of NADPH, which would not be used to regenerate the RuBP. So all of those things would lead to stopping of the light-independent reactions, which would mean that the plant would be unable to fix sugars. So what they were looking for here was there'd be no chemiosmosis, because there'd be no proton pump. It's going to stop photophosphorylation. It's going to produce less ATP or no ATP. It's going to produce less or no NADPH or reduced NADP. Therefore, there'll be no Calvin cycle because there's no regeneration of the RuBP. Won't be able to make any hexose sugars, therefore there's no respiratory substrate, therefore respiration ceases, therefore the plant dies. This question looks at comparing the effect of wave action on the height of the shells of a dog whelk, and they're comparing an exposed shore and a sheltered shore. So they're measuring the size of the shell height. They used a random sampling technique to collect 50 shells from an exposed shore. They measured the height from the base to the conical tip. They return the whelk to its location. They repeat the process for the sheltered shore, and they give you the results in a table. Now, what you can see from this table is that they've got the number that they have measured, and they've got a range, which is from the largest to the smallest. They've got calculated a mean. They've got a standard deviation, which tells you about the distribution of the data. They have a range for the exposed shore. They have a mean, which is very different, and they have a standard deviation, which is very similar. So they then look at a t-test. So they suggest that you carry out a t-test to determine the significance of the differences between the shell height on the exposed shore and the sheltered shore. They ask you to calculate the t-value using this formula. They give you the formula, and they also mean that you can then give you all of the things that you need to calculate that, because they've given you the standard deviations, so you just need to square them. They give you the mean values. The sample sizes you just need to calculate from working out how many they did, which remember was 50. So they use 50 of here, and there are 50 values here. So you just have to put the values into the equation and calculate the answer. So they give you marks here for showing the steps in the calculation, and then giving your answer to 2 decimal places. Now what you should have worked out was 13.61, and putting it into 2 decimal places is 13.61. The null hypothesis is that there is no difference between the means of the 2 shell populations. Critical values at 98 degrees of freedom are shown in the table. Your value that you calculated was 13.61, which means that the value is off here. It's higher than the critical value for p is less than 0.001. So using the table of critical values, so referring to this critical value here, explain whether the student would be able to accept or reject the null hypothesis as a result of the t value you calculated. So what they're expecting you to suggest here is that the probability is highly significant because the calculated t value is greater than the critical value for p is less than 0.001. Therefore, there is a less than 0.001 chance that the differences in shell height can be due to chance. Therefore, the null hypothesis can be rejected, that the difference between the shell sizes is due to chance, and that is probably due to some other factor such as the wave action. Now, the students organise the data into classes. These are the classes here. These are the size classes. You've got the sheltered shore and the exposed shore. And then you've got the totals for each one. And then plot the most suitable graph of the data given. Now, looking carefully at this, remember that there is an overlapping data set in that these two values overlap with these two. So what they're hoping for you to plot is that you would plot a histogram, because that would be the most suitable plot with these classes of 3 millimeters in size difference. So the answers that they're looking for would be that you would plot a histogram, and that histogram would be correctly plotted so that they would look carefully at the values that you had plotted and make sure you plotted them in the correct place. So you do have to plot these accurately. That the two sets of data need to be distinguished by using a key. That the x-axis is labelled height in millimeters, and that the y-axis is labelled the number of dog whelks. So along this side we're going to have millimeters, and up here we're going to have number. That it should make a good use of the graph paper, and that the axes are scaled correctly. Now, when you've got these two sets of data distinguished by a key, remember that these values here and here are going to overlap, so that you would have to have, for instance, for 23 to 26, that there would be a value of 5 for the sheltered shore, and that there would be a value of 12 for the exposed shore, and the same with the values for 27 to 30. So now they expect you to use the data and the graph to discuss any correlation between the height of the whelk shells and the type of shore, and then explain why there would be that correlation that you have seen. So what they were expecting here was that you would mention that there was a positive correlation between the height of the whelk shell and the type of the shore, that the more sheltered shores had bigger whelks. If you were keen, you could have measured a correlation coefficient. You would see that shore action from the data has an impact on height, and that there's an adaptation to harsher wave action is that they are smaller. Now, on to suggest a limitation of the procedure that you use to gather the data and recommend how you would improve that. So you've got to link the limitation to your recommendation. So first things that they could have expected you to put for this was that there was no detail given of the random sampling technique, and how you should carry out random sampling would be to use coordinate tapes and randomly generated numbers to carry out random sampling with quadrats, so that you may not have randomly sampled them appropriately. The next thing you could have done was specified which measuring instrument you used, and the solution to that would be to use vernier calipers and then say that what precision it should have, which should be more than 0.5 millimeter or less than 0.5 millimeter precision. Okay, next you could have incorrectly identified the dog whelk, because there are several species of mollusks that are similar to the Nucella. So the solution to that would be use an identification key to correctly identify the whelks to make sure you're measuring the correct organisms. Finally, the classification of the shore was subjective. So you could have used a method for classifying the shores into sheltered or exposed. So all of these, a limitation and then an improvement, and that gets you the 2 marks. How could the students improve the accuracy of their data? Well, they could improve the accuracy by increasing the number of Nucella used, by doing more replicates to to do the entire experiment again, and if you had more data, you would have the more data you gather, the more accurate it's going to be. Discuss the validity of the conclusions that you've made during the experiment. This is give arguments as to why it might be valid or not be valid. So you could say, first of all, that it's not a valid conclusion because you only sampled a small percentage of the nucella that were present, which could lead to skewed data, that human interpretation of the measurement causes accuracy of the data to be questioned, and that there might be genetic variations or subspecies, and that's not taken into account. And then why it might be valid was that you did use a random sampling technique, that you did use a large sample size, and that you gave instructions for consistently measuring the size of the shell so that your data is accurate. You did do sufficient random sampling, you did do enough samples, therefore the conclusions that you have reached have been valid because of those reasons. Botulism is a condition resulting from the action of the botulinum toxin. Main symptom of botulism is skeletal muscle weakness, which can be fatal. The toxin is produced by the anaerobic bacterium Clostridium botulinum. What does the information anaerobic suggest about the bacteria? Well, this suggests that it can only respire in the absence of oxygen. They are crucially looking for you to say that it only respires in the absence of oxygen. Not that it can respire with oxygen or without, but that it only respires in the absence of oxygen, and that the presence of oxygen would prevent the growth of that bacteria. So the toxin is initially produced as a single large polypeptide that has a low potency. After the toxin has been acted upon by a protease. So a protease is something that hydrolyzes proteins. Two chains are produced which remain connected by a disulfide bond. The disulfide bond, remember, is between two amino acids. In this form, it is far more toxic. Describe the action of the protease when it acts on the toxin. Well, the protease is going to hydrolyze a peptide bond, but the two chains are going to remain connected together by the presence of the disulfide bond. So they're looking for you to crucially say it hydrolyzes the peptide bond between the two amino acids which are joined by the disulfide bond. So next, an assay using 99 mice was used to determine the median lethal dose of the toxin. Suggest what is meant by the term median. So median means the middle value, and the middle value with your 99 mice will be the quantity of toxin required to kill the 50th mouse when you line them up in order of the lethal dose. Fairly unpleasant for the mice, but that's the value, which is the amount that you require to kill the 50th mouse when they are ranged in the order of lethal dose. Now, the median lethal dose of this toxin is in the range of 5 to 50 nanograms per kilogram of body mass, depending on the toxin type and the method of introduction. Calculate the probable lethal dose of the least toxic botulinum toxin for an individual with a body mass of 85 kilograms. So the least toxic will be the 50 nanogram. So you need to take 50 nanograms and multiply by 85, because that's the body mass, and this is per kilogram. And that gives you 4,250 nanograms. Now we need to give the answer in micrograms, so the answer is 4.25 micrograms. And just to confirm that, there's the answer from the examiner, 4.25 micrograms. So now they tell you about the mode of action of the botulinum toxin, and they say it acts primarily at the nerve terminals of stimulatory motor neurons. Part of the molecule binds irreversibly to specific receptors on the presynaptic membrane. The complex is then taken into the cytoplasm of the neuron, where the disulfide bond is broken, releasing the section of the molecule which acts to block the release of the neurotransmitter. So if you block the release of the neurotransmitter, then there'll be no postsynaptic stimulation at the neuromuscular junction, And they ask you then to explain why botulism can be fatal. Now, fatal is hugely important to this answer. So any explanation must include why that is going to kill the organism. So they're looking for here things like the intercostal or the diaphragm muscles are paralyzed, that they don't contract. And as a result, ventilation, which is the movement of the respiratory medium, which is the air over the respiratory surface, which is the alveoli, is reduced, and that the oxygenation of blood is compromised. Therefore, named cells or a named organ cannot obtain oxygen for respiration. Therefore, the organism dies. So it must refer to why this would be fatal. So now it goes on. There are a number of different strains of the botulinum bacteria. Different strains produce immunologically distinct forms of the toxin. Explain, tell us lots about the theory, why the toxins produced by different strains are described as being immunologically distinct, and how they will be dealt with by the immune system. So, first of all, it's a 6-mark question. In order to put you into level 3, which gets you 5 to 6 marks, you need a full explanation of why the strains are immunologically distinct, and you need a description of more than one method of action of the immune system. To put you into level 2, you needed a full explanation of why the strains are immunologically distinct, and an attempt to describe a method of action of the immune system, or a description of more than one method of the action and an attempt to explain why the strains are immunologically distinct. So, why are they immunologically distinct? It's because they are genetically different and will be producing slightly different toxins. So each toxin would have a different tertiary structure as a result of a different sequence of amino acids, as a result of the DNA molecule coding for that sequence being different. It's crucial to point out that the toxin would act as the antigen. And remember, the immune response is determined by the shape of the antigen in terms of clonal selection of the B and the T lymphocytes. Different compounds, i.e. different toxins, will have different shapes. And then on to explaining the effect of that on the immune system. Well, remember that you've got antigen presenting cells which would ingest the antigen, which is the toxin, and display the antigen on their surface. That there'd be an interaction between the antigen presenting cells and T helper cells, that the B cells will be activated by the production of the interleukins by the T helper cells, that there would be clonal selection and clonal expansion of the correct B cell, which has the receptor which is complementary to the antigen being displayed on the surface of the antigen presenting cell, that the B cells would then differentiate into plasma cells, and the plasma cells produce the antibodies. Those antibodies bind to and neutralize the toxins. This is the insert for the next question, and the next question is about termites. And termites are insects which are social insects, and they live in colonies, and they live in colonies at the base of the termite mounds. And you should be able to see that there are spaces within the termite mound. Now, there is a comparison of 2 termite mounds. These ones are these ones here are in Queensland, Australia, which is, it tells you in a question later, some 3,000 miles south of the equator, and these ones are in Africa. Now, what you should be able to see is that the orientation of these is that they are flat and thin, and that's referred to in the next question. So, termites are highly social insects. They're thought to have evolved from earlier forms of insect at least 150 million years ago in the Jurassic geological period. They're related to cockroaches. How might scientists a century ago, this is absolutely crucial, have known that termites evolved in the Jurassic geological period? Well, they would have looked at fossils, and they would have seen termites in Jurassic geological period rocks, and they would not have seen termites in rocks that predated that period. So the answer they were looking for you to put here was that they would see fossils in Jurassic rocks. What new source of evidence might help today's scientists, as technology has changed and improved, to find out how closely related termites are to cockroaches? So the new source of evidence might be the use of biochemical evidence such as DNA or cytochrome C. So by DNA sequencing and comparison of the genomes of termites and cockroaches, and by looking at the amino acid sequence of cytochrome C in both termites and cockroaches, you would give you an idea of how related they are. Figure 5.1 on the insert that we looked at previously, termite mound, approximately 1 million individuals, taken in Queensland, Australia, which is about 3,000 kilometres south of the equator. The interior of the termite mound is full of interconnecting chambers. At the top of the mound, some of the chambers are open to the air outside. Worker termites spend all their time working in brood chambers low in the mound, where the eggs and the larvae develop. Explain, tell us the scientific theory, why, how carbon dioxide produced in the respiring body cells of the termite workers is removed to the air outside. So this is a 4-mark question. There are 2 parts to it. Firstly, how is the carbon dioxide that's produced in Krebs cycle and link reaction in the mitochondria of the cells of the worker termites, how does that carbon dioxide leave the termite? And it leaves it by diffusion, because it diffuses down its concentration gradient into the blood of the termite. Well, they say blood here, I would disagree with that, but there we go. Carbon dioxide diffuses down the concentration gradient out of the respiring cell, carried through the body from the cell to the tracheoles by the blood, and then the carbon dioxide diffuses out through the tracheoles and then through the spiracles. Well, having left the termite, how does it get from the base of the termite mound to out of the top of the termite mound? And that is through convection currents, because respiration generates heat, the hot gases expand, so they rise up through convection to the vents at the top, and then the carbon dioxide leaves. Finally, in Africa, close to the equator, the mounds built by some species of termite are blade-shaped. So they're referring to, they are this sort of shape if you looked at them from the top, and you can see that on the picture on 5.2, and with the long axis pointing north to south. So this suggests why the African termites need to build mounds in this shape and orientation. Well, firstly, why do you need to build them in this shape? In this shape, it means that because they are blade shaped, they're going to have a very large surface area to volume, which is good for the dissipation of heat. And why in this orientation? It's to minimize the amount of the termite mound which is exposed to direct sunlight. So the shape is due to giving you an increased surface area to volume ratio, and the orientation is so that the smallest area is exposed to the greatest heat. Most termites only eat dead vegetable matter, so their principal food source is cellulose. Now remember, cellulose is a polysaccharide, and it's a polysaccharide of glucose, and it's a particular form of glucose which has the OH on the 1 carbon, which is up, and on the 4 carbon it's down, which means that in order to form a glycosidic bond with the next one, it has to be the other way up. So it would be like this. So then you form long straight chains of alternating beta glucoses, and those long straight chains form hydrogen bonds with other long straight chains, and that gives the cellulose a tensile strength, and that tensile strength enables it to be extremely good at resisting the expansion of the cytoplasm and therefore osmotic lysis by the plant cell. So state the name of the monomer. It is beta glucose. Termites, such as the species that built the mound on the insert can be classed as keystone species. Use the information given about what the termite does, that it eats only dead vegetable matter, give the information given to state one argument that supports this statement and one argument that does not. So, firstly, why they're an important keystone species is that they are nitrogen or carbon recyclers or saprotrophs, and the other one is that they are not a predator. There's lots of them, they have a large biomass, so the effect on the environment is not disproportionate as it would be if they were a keystone species.